Hi everyone. In this series of videos, we've explored the immune response following exposure to an antigen. In this video, we'll look at the specific example of HIV infection and how it affects the immune system, potentially leading to the development of AIDS. First, we'll look at the structure of HIV, or to give it its full name, human immunodeficiency virus. AQA requires you to know the general structure of a virus and also the specific structure of HIV, so let's see how they compare. All viruses contain genetic material, which may be DNA or RNA. You need to remember that HIV is an RNA virus, and for that reason it also contains an enzyme called reverse transcriptase. Reverse transcriptase produces a DNA copy of the viral RNA genome, allowing it to be integrated into the host cell DNA. The genetic material of viruses is protected by a protein capsid, and we find this in HIV too. The final component found in all viruses are attachment proteins, which are used to bind to receptors on the host cell surface, enabling the virus to enter. In the case of HIV, the host is a helper T cell. The HIV attachment proteins are displayed on a lipid envelope rather than directly on the capsid. This lipid envelope is formed from host cell membrane, as we shall see next in the process of HIV replication. The host cells for HIV are helper T cells. These cells have a receptor on their cell surface membrane. You may hear this referred to as a CD4 receptor. The attachment proteins on HIV are complementary to this receptor, and so when they bind the receptor, the protein capsid and its contents, including the RNA genome, can enter the helper T cell. Once inside the cell, reverse transcriptase produces a DNA copy of the HIV RNA base sequence, and this is incorporated into the host DNA. Viral proteins can now be produced, and new viral particles are assembled. The new viruses leave the cell in a process called budding, as they become surrounded in a phospholipid bilayer of host cell membrane, forming the new viral lipid envelope. On initial infection, the person often experiences flu-like symptoms, similar to the symptoms of other viral infections. During the second stage of infection, HIV can lie dormant in some cells, but helper T cell numbers are gradually depleted. An AIDS diagnosis is made when the number of helper T cells falls below 200 cells per milliliter of blood, and disease symptoms of opportunistic infections occur. Nowadays, the progression to AIDS can be slowed using antiviral drugs. However, there's currently no vaccine against HIV and no cure for AIDS. In the next video, we'll consider some of the modes of action of antibiotics and why they cannot be used to treat viral infections.